the head of healthcare at Edison Group. I have the distinct pleasure of speaking with Nicholas Waters from IRLAB.
Thank you, Sue, and thank you for having us today.
Great. Let's kick off with some questions here. Okay, great. So IRLAB seems to be taking a comprehensive approach to the development of Parkinson's disease. Could you give us a little background on that?
Absolutely. Parkinson has been a part of my and my colleagues' life for the best part of 30 years now. The team in IRLAB all have a history together with Professor Arvid Carlsson, the scientist who discovered dopamine in the brain and levodopa as a basic treatment for Parkinson's disease. So that's where we come from, and we were raised in his laboratories. And when we started IRLAB in 2012, we saw a huge opportunity in developing a number of compounds addressing specific issues for people living with Parkinson's disease and of course, their caregivers as well. So IRLAB is founded on the understanding that there is a huge unmet need in this space, but there is also an opportunity to grasp.
So can you inform us on your approach? I think there's an ISP platform involved.
Yes.
Does that mean machine learning?
Indeed. We started developing a phenotypic discovery strategy, or systems biological is a more popular term these days, in the early 90s. In the early 90s, we were working with programs which were asked of us basically from the industry. That was a university industry collaboration period of our careers. And at that time, we discovered that many times the candidate drugs that were selected in a certain campaign were not the right ones. They were selected based on certain criteria, certain simple criteria. So at the time, early 90s, we started to discuss how could one devise a discovery strategy which in itself would support serendipitous findings. So planned serendipity was a kind of a term that we threw around in the offices and the laboratories. After that, we started to structurally document the effects of all CNS classes. This is a comprehensive strategy to map out what we call the property space of all CNS classes, and the most prominent and the least prominent representatives of each of those classes. It's been a hard work, tedious work. We've collected in vivo data describing each molecule's effect at the system level with wet laboratory data. So we collect behavioral data, we collect neurotransmission related data since it's the brain, and we collect gene expression data to understand which processes are triggered by different treatments and also in pathologies. This means that we can create pathological states in our laboratory, which we then can look for drugs, treatments that actually reverse those errors that occur at the system level. And that's been extremely helpful, especially in the PD space for us. It sounds like a simple disorder. You lose a function and you just try to replace that with something that creates a better function. It isn't that simple. It's a system disorder, which we have tried to address. So in our pipeline, we have developed products or compounds or programs for each and every complication that occurs during the course of the disease development in PD.
That's actually super interesting. I mean, it seems like there's some new developments emanating from this ISP platform, including IRL 757, IRL 942, and PU003. Maybe it makes sense to kind of delve into those a little bit?
Indeed. If we put aside our clinical assets right now, which are in phase 2b, that's mesdopetam, which we've licensed to Ipsen, and pirepemat, which is now in a globally huge, a large study addressing falls in Parkinson's disease. But if we look at the preclinical assets that we have, that have come out from the discovery using this machine learning strategy that we have developed over the past 30 years. 942 is a drug which is designed to improve cognitive function across neurological disorders, actually, not only Parkinson's disease. The data we have generated so far is extremely promising in terms of the breadth of cognitive enhancing properties that we see with this molecule. At the same time, the ISP system have supported us to tweak the profile of the class of molecules we're working with there, 752, that's pirepemat, 942, and 757 as well. We have tweaked 757 a little bit towards the effective direction. That is effective, emotional, and let's call it also cognitive direction. So we have small differences in the profiles of these molecules, but they have that may have huge impact on the outcome in clinical trials. And that is directed by our machine learning strategy.
Great. I mean, could we delve into your two leading assets, maybe look at some key milestones if possible?
Absolutely. In the mesdopetam program, which is a drug designed to treat both dyskinesia, which is a syndrome inflicted by the use of levodopa, but also a consequence of the disorder. So there's interaction. So it's a drug which treats dyskinesia, but also has a very powerful antipsychotic properties, at least in the preclinical setting. The dyskinesia has been investigated in phase 1b study and in phase 2a study. And after that, we have now launched a phase 2b study, which we are completing this year. So next big milestone in that program is the top-line data from this international trial, which is run right now in the US and Europe.
It appears that you're very focused on mapping the progression of Parkinson's disease.
Yes.
Does your platform give you flexibility to expand into other areas of CNS?
Absolutely. If one goes back in history, we had a very strong progress in developing novel types of antipsychotics in the early 2000s. We had a quite proliferative collaboration with Merck Sharp and Dohme in those days, but also a collaboration with Astellas in Japan for antipsychotics. So that's a thing that's in the past for us, but we do have ideas of how to design and develop novel strategies for psychosis. And that comes to a large extent also knowledge that we've created using looking at mesdopetam, which has an antipsychotic profile which is novel in PD, I should say, but we can translate that likely to other types of psychosis. And pirepemat also has antipsychotic-like properties, but with a totally new mechanism. And we're trying to ride on that a little bit. So long story, short answer, yes, we can move into depression and psychosis and other types of neurological or psychiatric complications.
Great. So, just as an investor, like what should they look out for in the next couple of years? Maybe we can look at two buckets, maybe in the next 12 months and then maybe longer term.
I would say the readout from the mesdopetam trial would likely be the most interesting thing for investors in our portfolio this year. And then following that is, of course, the initiation of phase 3 studies, which is something that's in the hands of Ipsen, our partner in this program. Next thing there would be indication expansion into other important indications with that molecule. For pirepemat, that is concluding the study. That's the following year in 23. We expect to have data late 23 with pirepemat. In the meantime, we almost have a, let's call it a traffic jam in terms of moving things into phase 1. We have 3 programs now, the ones we talked about before, preclinical assets 942, 757, which will reach phase 1 in this time period in the next 24 months. And in the P003 program, which is a highly interesting program where we're looking at solving a problem in the treatment of Parkinson's disease, which have been an enigma since 1969. And we have kind of revisited that problem. And that's a long-lasting levodopa-like, but not based on levodopa at all, but to have a once daily treatment for the basic symptoms of Parkinson's disease. That's something that has been an enigma to achieve without levodopa, but we think we are on a path here to find something.
Well, it's always a pleasure to meet with you, Nicholas. So I look forward to speaking with you again soon.